Entomological Society of America meeting here in Knoxville, Tennessee. I'm Crystal Ernst. It's about time I met you people. This is Morgan Jackson. Of course, you know him very well.
Yep. Hey, everybody. So we're in Knoxville, Tennessee at the biggest entomological meeting that happens every year. So this is the ESA is how we usually go about talking about it. And this year, I think I've heard numbers about 3,000 people turn on it. It's going well, at least so far. Crystal's given her talk already, and we're going to introduce that in a second so everybody can hear what Crystal does and get to know Crystal because she hasn't been on yet. You haven't had to stare at her or listen to her yet, as you have us. So, but anyways, so Crystal, well, with that segue.
Yes.
So what do you do, Crystal?
So I am a PhD candidate at McGill University, which is in Quebec, Canada. I'm just about to hit my 3-year mark, so I've been doing this for a little while. Previously, I did an undergrad and a master's degree in biology at Carleton University, which is in Ottawa, Ontario, also in Canada. These days, you can find me mucking around in the Arctic. I study mostly sort of food web ecology of terrestrial arthropods, and I'm particularly interested in beetles, so that's most of what I talk about these days.
Sweet. So that's your academic cred.
That's my academic cred.
What about your your non-traditional academic cred?
So the things I do online, in other words. According to my name badge here, I tweet. My handle is @geekinquestion. That comes from my sort of little, it was once a pseudonym, but I'm now out of the closet, so to speak. So TGIQ is how I'm known elsewhere online. I've been running a blog called, there was an earlier permutation of it called Fall to Climb for a while, but about a year and a half ago, I guess, it changed to The Bug Geek. So buggeek.com, that's where you can find me. I like to talk about bugs and pictures about bugs and a bit about research and a bit about grad student life. Gosh, what else do I do? Morgan and I work on the, it's a little embarrassing. Morgan and I work on the ESC blog together. And I've got a couple of YouTube pages. And
yes, definitely check out Crystal's YouTube page, which I assume you can link to from your blog.
I can probably, I haven't, and I probably should. Yeah, it's mostly a sciencey dissection-based blog, so it's mostly intended for zoology students, so high school or undergrad zoology. So if you want to see shark guts and starfish guts and fish guts, and I've got all those things on video. So yeah, those have been a lot of fun. Yeah,
and you're like super famous on YouTube. How many hits? You said this last weekend.
Oh my, I don't even remember. I have in a matter of 3 months, I had 100,000 page views or video views. I don't even know how the metrics work yet because I'm still so new at it. But apparently, people are really excited about guts of things. But it's interesting because I'm getting a lot of people, high school students and high school teachers and undergrad teachers who are using, and even homeschool teachers that are using these in classroom settings.
That's awesome.
So they seem to be useful tools, not just for my students but for other people. So that was a good investment. And
that's huge.
Yeah, it was pretty fun.
So I'm going to keep, because we've never actually talked about this aspect of it for all of this amount of time that Crystal and I have been hanging and interacting.
Yeah.
I've never actually asked Crystal about the YouTubing. So how did you end up going about it?
So I've been TAing this intro zoology lab for I've done it for 3 years now, and in the second year it underwent sort of a major change, and the way the labs were laid out really changed, and the amount of students I had in the labs got huge, and so I had about 40 students in a lab at a time, and when you're doing dissections, it's always I can't find this thing, and I don't know what I'm looking at, and I was just spending all class running around like crazy trying to answer everybody's questions, and it just wasn't very efficient for me or for them. So I came up with this idea last year to pre-record dissections before the labs. So the students had them as part of their course notes, so they could go and they could check it out before the class, and then they also had them available, when they were studying, kind of thing. So I just picked out the animals that were the most common things that we were going to work on, and I just took my little point-and-shoot camera, and I had this little tiny tripod, and stuck it there with, just over the dissection trays, and I just talked through the dissections and all the different parts. And so I think I've done 14 or 15 different videos. So
pretty much the entire syllabus for an intro zoology.
Yeah, I start with, I think the first one I did was, the roundworms, and I go through the echinoderms, and then I get into, I don't even remember what else now. There's lots of vertebrates in there, so I do fish and rats and a pig and a shark and frogs and So, you know, and all the other little bits at the beginning of that. So, and I'm hoping to add a few more this year. We'll see. I've been getting requests now from other people. Can you do jellyfish? Can you do a cow eye? And it's like, well, I won't be doing that one. But I'll probably add a jellyfish and maybe a few other things this year. So
Sweet.
And I feel like the students love them on the course evaluation. People were really happy about those. So it was great. It was a good tool for me because then I could be like, go watch the video on your own time. You can pause, record, and so much more practical for the class. So yeah, so it was a cool venture.
Good.
Yeah.
So what are your opinions been of the meeting? So this is your first ESA.
This is my first ESA. I've been sort of doing the conference circuit this year. So
winning the conference circuit. We should point this out that Crystal is batting 1,000 for her President's Prize and competitions for her speaking. So she won the regional contest in the Entomological Society of Ontario. She won the big shebang there. Then last weekend in Edmonton, Crystal won pretty much every prize that was available to her. 2 photo awards. She won a big scholarship from the ESC for her research, and then she also won the President's Prize for her category. So we're
parasites. We should talk about this.
Yeah, we're definitely going to talk about Gordian parasites here.
Those are pretty cool. So so thank you for embarrassing me.
That's what the podcast is for. Now it's recorded for all time.
That was super. So I've attended these meetings. So I've gone from sort of the small regional one, and now I'm at this crazy international one. So I think that what did the ESO had about 180
ESO
people.
was about 60 people. I
Okay,
think.
so 60 people, so then about 300 people in Edmonton, and now this. And it's a lot to take in. I spent probably, I don't know, at least 8 hours in total just trying to get through the program to figure out what I was going to see. It's been interesting. I'm finding it difficult to make a schedule work that I can actually see all the stuff I want to see. But it's cool to have so many people from so many different fields. Although I have to say, as an ecologist, there's not nearly enough ecology going on here.
No, exactly.
Where is the ecology?
For the ag.
Yeah, and
it's
you could tell right away, I think, walking in, that there weren't going to be a lot of ecologists because there was a distinctive lack of polar fleece and cargo pants.
And sandals.
And sandals and sneakers and that sort of thing. So I broke the mold today because all the fancy students are wearing fancy suits and I'm wearing my blue jeans to present.
Like a true entomologist.
Like a true, I don't need to wear fancy shoes and pants to do entomology. But anyways, I'm judging now. I should stop that. But yeah, no, I think I've met a lot of interesting people, and I feel like I've made a few good connections, and it's been interesting as a Canadian to talk with American entomologists and American students to hear about, I'm going to eventually be looking for a postdoc in the not too distant future, so hearing about opportunities and funding and the weirdness of moving to the States and that kind of stuff. So it's been good for that.
Yeah.
So what do you think? What's your general impression of the meeting so far?
Oh, it's been good. As hectic as always. Like, this is my 3rd ESA now.
Yeah. You don't have the.
No, I don't have a fancy new ribbon. I got
new
nothing.
Member here.
I got nothing. I might go steal some.
Pro.
I've seen some people with ribbons saying my ribbon is better than your ribbon.
Really?
I want one of those actually.
I'd like that ribbon also.
Yeah, I think they're down in the lobby. We should go steal one.
We're gonna go get these ribbons later.
Yeah.
But sorry, carry on.
Anyways, so this is my 3rd one. And it's the same hectic pace. Like, the first time, you're just completely mind-blowingly overwhelmed by all the entomology going on at all times and all days, all times of the night. So
it's true.
Once you get through that first little triage and realize you can't see everything, and then you just start picking and choosing, and then you just start randomly sitting in sessions, then it's all good. It's just a little bit of a slight learning curve, but once you get it, it's a really sweet experience, and I recommend that anybody studying insects goes to something like this.
It's pretty cool, and I have to give props to the people who have organized it, because so far I found everything has been really smooth.
Yeah.
The facilities are good, and the volunteers seem to know what's going on, and
for sure.
no technical, I think this is the first time I've given a talk where there wasn't some kind of technical malfunction with the pointer or the PowerPoint or something. Everything has been, I have to give them a huge thumbs up for making our lives a lot easier as presenters. So
definitely.
Yeah, very nice that way.
Yeah.
And the other thing that's been neat for me here is running into people. We were talking about Twitter and all that, running into people from online. So there's been a lot of fangirling going on. We've met the bug chicks are here.
Yeah, the bug chicks are here.
People.
We're actually going to take the camera. We're going to talk to the bug chicks later, so watch for that.
Yeah. So a nice way to connect with people that we've been talking to online virtually for all this time. So
putting a face to the avatar is pretty big. So
exactly. So
but yeah. So anything else for now, or we'll cut off now and we'll come back later with some new guests. And we've been scoping out some people with some cool talks and cool science, and we're going to be dragging them onto the podcast. So I hope you like it.
Yeah.
And again, Crystal's here and she's here to stay.
Finally, about time. Sorry guys, it's been well overdue. So.
So we'll talk to you. So welcome back to Breaking Bio. So we've got our first volunteer slash victim here. This is Michael Naglee from Brigham Young University in Utah. And he's a third year, or if you're not from Canada, a junior. And he's here presenting some research. So we're going to talk to him a little bit about that. So what's your research on, Michael?
It's on earwigs or Dermaptera, and particularly looking at the parasitic lineages within it. There's 16 species that are parasites, or two suborders.
Sweet. So when you say parasitic, you mean like ectoparasites?
Yeah.
Like, so I think at some point in your talk you had the really hilarious slide. I laughed a lot, actually. I think I was the one laughing out loud that was louder than everybody else. But you had these earwigs are kind of like the love child of a flea and a louse.
Yeah.
This is essentially what you've got. So this is an earwig that's kind of weird.
One of them is about 5 millimeters long, and it lives off of rats in Africa, the giant rats, the ones that come up to here on your arm.
Crazy.
And they have specialized legs that allow them to cling onto the rats, and mouth parts that let them eat the dead skin and fungus on the rats.
Okay, so they're not actually sucking blood. They're just like picking stuff off of.
Yeah,
Oh,
they're actually
okay.
beneficial.
I was just going to ask, so do they harm the rats in any way?
No.
So can you technically call them a parasite then, aren't
That's
they?
why they're called ectoparasites.
Okay, so that distinguishes them from the harmful type.
So they're like the little feeder wrasse that you see on coral reefs and stuff, on rats, giant rats.
Yeah.
That's pretty sweet. Oh. Well, that changes. So you got giant rats and creepy, well, not so creepy, parasitic cleaner earwigs that
Yeah.
just live with them.
And
Oh, that's really cool.
do you,
There's also a group that does the same thing with bats in Malaysia. They're a lot longer though. They're about 25 millimeters instead of 5.
That's a really big earwig.
Yeah.
And they just, so how big are the bats that are on? Like, are they like
They
half the length
they
of
aren't
bats?
as closely associated with the rats. They don't like ride on the rat or
Oh.
the bats. They're mostly found in the caves, but they
Oh.
do eat the skin and skin glands.
So they really are like the coral reef. They just hang out, and then when the bat comes up, they climb on and kind of comb through, pick out
Yeah,
some stuff.
the relationship between them isn't super well known.
Oh, okay.
So, still trying to figure that one out. There's only 5 species of those,
Oh,
the bats. So,
that's really sweet.
so
Yep.
you found those ones, that was in Malaysia, you said? And the ones on the rats were in?
Africa.
Okay, so can we find anything cool like this, like up here in North America? Does that happen?
No, not that I know of.
Yeah.
Sorry.
That's a shame. Is there anything like even remotely similar? I've never heard of earwigs. I had no idea that earwigs did this. When you were talking about parasites, I thought you meant like parasites on the earwigs. But so, are there any other groups of earwigs that act in this bizarre kind of way, or anything?
Just those
Wow.
16 known species.
How did you find out about them? How did you get into this?
I actually was told about them.
That works.
And so that's how I found out about them. And then we had someone send the species to us, so
So
I
you got
didn't
collaborators.
actually get to go out. Yeah.
Not yet.
Not
So you're
yet.
still an undergrad. So if you were to go on and do grad work, would you want to keep doing this, or you want to move on to something else?
I'm not sure what I would want to do for
I have a
grad work.
sec-
I'm still figuring that one out. I have 2 years to figure it out. So
Yeah, fair enough.
Yeah. You're way ahead of the curve on respective on many
Yeah, I
undergraduates.
don't think I was giving talks at international conferences in my second or third years.
I was struggling with midterms in my second and third
Yeah, I
years.
think
I
that
was not
sounds
even in
about
a
right.
lab. So.
I know.
I'm still doing that too. I still struggle
Yeah,
with
okay.
midterms.
Well, it's amazing.
Yeah, but
that
you're
able to-
you're doing research on top of it.
Yeah.
That puts you in superstar category in my mind. So that's pretty awesome.
Well, thank you.
Yeah.
So what have we'll just do a quick wrap up. What are your thoughts of Knoxville this year?
I like it. I like learning about other people's research. I like looking at how science is moving forward. It's fascinating to me.
Mm-hmm. Sweet. Yeah, it's a pretty good meeting that way. So, anything else you want to tell the world?
No?
The world being our small viewership. No, that's it? Awesome. Well, thank you very much for stopping by and talking to us about all your work and cool, crazy, and not as scary as I had thought ectoparasitic earwigs. So I'm thus terrified now. So, anyways,
No
thanks.
problem.
Thanks very much.
That's awesome.
Thanks.
We'll see you around.
Okay. Hey, everybody. We're back again with our next victim. Is that what we're calling them now?
I think so.
This is Sarah Laboda. She's actually one of my lab mates at McGill University, and Sarah's been working on an amazing master's project, and she's here to talk about something that is very near and dear to her heart. And what is that, Sarah? Spiders. And why don't you just introduce us? Who do we have here, Sarah? This is your new pet.
Yeah, because we are still in Knoxville at the ESA, and there was like BioQuip, I think,
Yes.
that sells some live spiders and scorpions, and I fell in love with him or her. We don't know yet.
You have to wait for it to grow up a little bit more still.
Yeah, because I want to call her Dorothy, but maybe for a man it's kind of awkward. So
Yeah,
we gotta wait.
we'll have to wait and see. So so in addition to buying spiders, which are particularly awesome by the way, at conferences, what do you do in terms of spider stuff in your sort of day-to-day researchy life?
Life.
Yeah, or lack thereof, being research and all. Tell us a little bit about your work. What do you do?
I studied Arctic spiders with you actually for the project, the Northern Biodiversity Program. That was we went on the field in 2010, 2011 to collect some arthropods with pitfall, pen trap, melisy, and some sweep. And I was the one working on spiders, and I was working on patterns of diversity. So where we can find the species and why.
So I think a question that a lot of people will maybe have is like, really, there are spiders in the Arctic? Like, how far north are you finding these spiders?
So the northern we get was at 81 degrees, so almost like 800 kilometers from North Pole, and we found more than 3,000 spiders within 10 days. So I guess
3,000 spiders, like individuals, in 10 days?
yeah.
That's a lot of freaking spiders.
Yeah, so it's I think it's way more than people think. And when you go in the Arctic and you walk, you just can see all the spiders. running
It's just a
around.
carpet of spiders. There's no tundra. It's just spiders.
That's kind of what it looks like. Yeah.
Wow.
Yeah. So like, so in addition, so you found lots of them, but like, are there a lot of different species, or what kind of things are you noticing as in your travels across Canada there?
So the northern side we went to, we found only 8 species of spider. But if you, for all the side across the Arctic, we found more than 35 species of spider. And if we count also the subarctic area and more like a North Boreal area, we had more than 300 spiders.
So we're looking at neotropical spiders. We'll get back to Arctic spiders. So you said there were 300.
I identified 300 spiders, but it was
300
just
species of spiders.
Species of spider.
Yeah.
But we just look at ground-dwelling spiders, so maybe there's more.
Okay,
to
so
discover.
so you got ground dwelling. So what other what are the other group? Like, what do you mean? What?
Ground-dwelling means that they just run, so we can't find them in the traps because we didn't look at them actively. So maybe there's some that would just stay on the little shrubs in the Arctic that we didn't catch. So there's probably more than to discover there.
Cool. So you guys are looking mostly at like the cheetahs of the spider world, the ones that run across the ground to hunt, rather than, the stereotypical with the webs and the Charlotte's Web kind of orb weavers and stuff like that.
Yeah.
Are there any of those up in the Arctic? Because there's not a whole lot of vegetation up there.
Yeah, I don't think there's orb spiders in the Arctic, really north in the Arctic. Probably south, but
Where
yeah.
there's still shrubs and stuff.
Yeah.
There's not much for them to build things on, like build webs on, really. In those
Yeah, but
higher
we found a
regions.
lot of ligniphiidae, so dwarf spiders. Kind of cool, huh?
Wow.
Welcome to Breaking Bio, guys.
That's
All right.
Uh,
Well, we'll just sign off from the dark here. And we'll see how it goes. So if you couldn't tell, we're stealing space at the moment. So
yeah. Thank you for showing us your friend, and thanks so much for talking about your Arctic spiders, Sarah. And maybe tell you, look, we were telling one of our other guests, maybe once you've got a paper out on this, because there's some cool results in this study. Maybe you could chat with us again and tell us a little bit more about what you found.
yeah. Thanks.
Perfect.
All right,
Thanks
thank
to
you. Spider says goodbye.
Hi, so we're back here with Matan Shalomi from the University of California at Davis. And so Matan is an entomologist, but we're going to be talking about something that he did this summer that is a little bit out of the ordinary. But first, we'll let Matan discuss a little bit about what he does for a living. So, what do you study normally?
All right, so I'm a PhD candidate at Davis, and I study the digestive system of walking sticks, the Phasmatodea.
Oh, that's really different than I was expecting.
Yeah, it is,
having
too.
nothing.
Yeah,
All right, well, that's awesome. So, you're mostly like a morphologist.
it's morphology, physiology, bacteria kind of stuff, yeah.
All right.
Has absolutely nothing to do with what I'm about to talk about.
That's totally cool. I was not expecting that. That's even cooler. We can talk about that afterwards, actually.
That's
Sure.
pretty interesting.
So that's pretty
Yeah.
cool. Okay, so as you can probably elucidate this, we were not planning on talking about Phasmodia digestion, but actually Matan this summer published in the extremely prestigious Annals of the Improbable Research, which is best known for their Ig Nobel Awards. So they give out that crazy Ig Nobel Award and have the big ceremony and everything. But anyways, Matan published a really cool paper, and I'm going to bring it up here so that I don't mess it up like everything else I've done today. And the paper was titled A Phylogeny and Evolutionary History of the Pokemon, like the video game Pokemon. And so, how did this project come up? Because this is awesome, and I'm amazed that nobody has done this before.
So am I, honestly. People have attempted it online. You go onto like DeviantArt, you see little phylogenies, and if they're any good, it's usually someone took a real life phylogeny and just replaced the animals with Pokemon. But I figured you could do this quantitatively. You could make it a repeatable, actual scientific endeavor. I like Pokemon. I was growing up when Pokemania started and everyone was playing and watching the show and things like that. So it's always kind of stuck with me how just Pokemon is so much like biology. I mean, the guy who created it was an amateur entomologist. He liked insects and sort of created this entomology simulating game, only added Pokemon combat. So the idea was make a phylogeny based on the Pokemon, and since they obviously don't have DNA, the only thing I could find out similar was their attack sets and things like egg group and moveset, body type. If you're a Pokemon person, you'll know what those are.
Yeah.
Yeah.
I thought that was brilliant. Like when I was reading the paper, I'm like, so how did they do this? Did they just look at them? But then you went through and you looked through their stats, and I mean, that was really cool. It was essentially what I would consider a gene tree in many regards, is how these things are heritably passed on and such like that. So I thought that was brilliant. So, you got some really cool results here. We'll try. I'll actually buy the full res thing because right now it's just a little bit of a crappy
I can
GIF on
send
my
you
phone.
it's on Imgur or something.
All right, that's awesome. If you can send me the phylogeny, we'll show it up there in the
course.
middle of this.
Yeah.
So what were some of the cool things that you actually got out of this? Because this isn't just some random tree.
Yeah.
You actually got some really awesome structure out of it that makes and has some really cool parallels to actual real animal.
Well, that's the thing. I didn't expect the tree to work, but it it did. You plug in just their all these taxonomy. We use an actual phylogeny formula, Mr. Bayes, and all these other software. Ran it for about a day, 16 million generations, and we get this tree that just looks like a real tree. Pokemon life seems to have evolved in the water, just like our life. You start with the water types, and then terrestriality evolves from that. A lot of the different, I guess you call them clades of Pokemon, do seem to come together.
Yeah. So cat-type Pokemon.
What was I saying? Yeah, so the dog-type Pokemon all seem to come together, and the cat-type Pokemon are close together, and the fire types all evolve from within the dogs and things like that. And you get your transitional species, so you have Venusaur and Sawsbuck, the normal grass types, kind of look like animals with plants growing out of them. Those are the transitions into the whole plant type clade. So it works, which doesn't make sense because it's all fake. These are not real animals, but either intentionally or not, the people who were designing the movesets made it.
A true evolutionary.
It works, yeah.
So that's cool. So getting back to Venusaur, because I thought that was really cool that you had this really awesome transitional thing. So to bring this back to the insect world, could we possibly see a correlation between Venusaur and, say, some of these aphids that are incorporating, chlorophylls and algae and stuff like that? Do you think this is a symbiotic relationship that eventually spawned into this super successful plant-type Pokemon?
Possible. Actually, well, a lot of the Pokemon are based on real-life animals. For example, there's Paris and Parasect. Do you know those?
Yep.
I would call those cordyceps, personally.
Yep.
I mean, it's this arachnoid creature with fungi growing out of it. That's pretty cordyceps to me.
Yep.
And when you actually look at a lot of them, the animals, the fish, they all seem to match, especially if you know your Japanese animals. There's a lot more parallels, but you have Beedrill, which is very obviously the Japanese hornet, the giant one. The butterflies all seem to match these Japanese organisms. The guy who created all this, they definitely paid a lot of attention to biology, and there's a lot of real-life animal inspirations in the Pokemon, and it's really fun to find them.
Yeah, for sure. I thought it was an absolutely brilliant paper, and the fact that you got so much actual resolution that made sense and had so many correlations, I thought was just freaking awesome. So that just blew me away when I read that. So you guys need to be commended. So are there any future plans for the next step in Pokemon taxonomy and systematics? Are you going to revise, the fire type Pokemon? Because I know there's a lot of really weird names, and you brought it up in the paper about, some Pokemon were described off the males and some were females, and they're really the same.
That could be problematic.
Yeah, the whole Nidoqueen, Nidoking thing. There's, I mean, it's just like real taxonomy, which again is why this is all so funny. But like, you get like the splitters and the lumpers, and like you get these like the sexual dimorphisms that people misidentify as different species, and that seemed to have happened in the Pokemon phylogenetic history. And I talk about that a lot in the paper. I just try to. The whole thing is delivered straight face. It's like
it really is. It's awesome.
Yeah, like all these like problems in the taxonomy and the problems with youth using Pokemon as fighting and that's threatening their conservation and all these other things. It was a real pleasure to write it, and I'm just glad I managed to get this in. Is the fourth co-author. So I wrote this along with two other entomologists in my department, and the fourth co-author is Yukinaro Okido, which in English would be Professor Samuel Oak from the Pokemon games. We use his Japanese game because his name I figured that might go a little bit easier with the editors, but they pulled that in.
Because they were really worried about, science being done here and not making fun of science. You wouldn't want them to poke fun of too hard.
Oh, heavens no. And on some probable research, they published a taxonomy of chia pets.
Yeah.
They did...
you guys are way above that. Like, if you guys don't win the Ignoble, then I'm totally...
Well, the Ignoble is real research. That's the thing.
I fully consider this real research. This is testing phylogenetic concepts.
Now, I have a question, though. Have you been challenged on your results yet? Has anybody come out there and...
I have actually. Well, the problem is it violates Pokemon canon. Like, there are a few things here that don't seem to make sense within the storyline. I mean, I haven't gotten any, like, hate mail from the Arceus creationists, but...
That's good, that's good.
But I want to get this thing all spread, so I've tried to post it on, like, Pokemon forums and things like that, and some people do take issue with it. A lot of it is, I mean, there are some things that don't make sense, like what are the bats doing in with the bugs, Golbat, Zubat group. I mean, that's just long branch attraction, I can't help that. But no, there are some conclusions people don't seem to like, and I'm like, well, make your own phylogeny. I mean, I would love to see people writing responses to Annals of Improbable Research. It's like, no, this phylogeny is wrong, here we used, what do you call it, more neighbor joining things, and we got this, we got that. So I would like to see more research on this, but wouldn't that be a terrible waste of resources?
I think it's a fantastic use of resources. If it gets kids thinking about phylogenies and taxonomy, I think it's freaking awesome.
Yeah.
Well, and also I was going to ask, like, has any of this work become useful to you in terms of like your real research? Like, not that this isn't real research we've discussed, but yeah, have you used any of these tools or these approaches or anything? Has it become a useful exercise for you or for people in your lab, or is it really just for fun?
So the actual making the tree that was done by one of my co-authors, I gathered all the data, which was an enormous pain in the butt, but he ran the genetic analysis. What this has done is I work at the Bohart Museum of Entomology in California, Davis, open to the public Monday through Thursday. And we have gotten people coming
We'll put the website up, don't worry.
Oh, yeah. We have gotten people coming into the Bohart Museum specifically to see the tree. We have it all printed out and laminated. And people are coming in just based on this Pokemon paper. And it's like, well, we have other stuff in the museum. We have some live insects.
Some real things that you go over.
But sure, while you're here, here's my tree, and let me talk about it.
Well, that's actually a really cool way of, like, sort of tapping into sort of a more, socially, accessible phenomenon and like different age groups as well, and like drawing them into the museum where you can show them other science, right? Like that's a neat link that you're
how they
making
can do
there.
it for real for an actual career. Like you can do this for real with real organisms.
That's pretty cool.
There were a few like actual papers, including one I saw in American Entomologists about scientists sort of lamenting how kids are so excited about Pokemon, but there's this real world out there. It's like instead of like catching fake animals, go out and collect the real ones and study the real ones. How kids can memorize like 150, 400, or up to like 693 Pokemon and all their moves, but they can't really remember the Latin names of the creatures.
The seagull that flew past their window while they were playing.
Yeah, exactly.
Yeah.
So I can understand the lamentation, but at the same time, Pokemon is so much fun. I feel like baby steps. People will start with Pokemon. It's the gateway to the sciences.
I think that's a great
gateway game.
I know it was for me. I mean, I'm not gonna lie. I mean, I played Pokemon when I was a kid, and then I grew up and found out I could be an entomologist who goes out and collects and studies these things the exact same way. I'm like, I'm sold. I'm in.
I think I started with the opposite. I always want to be an entomologist, and I was like, wait, Pokemon is like entomology. Awesome.
I can start early. Sweet. That's so cool. So cool. So one last question, which I think we need to get under the wire is, what did your advisor think of you guys dinking around with Pokemon in the lab?
I was doing this on my off days in the weekends when I wasn't at work, so I didn't mention it. My advisor, Dr. Lynn Kimsey, is a fantastic person. She's really, really cool with me occasionally taking side projects, especially since they do eventually get published and bring credit to our department in some way. So she's very flexible, I think it is important for a grad student to get the perfect advisor who is what they want. And I liked having an advisor who lets me do these silly little side projects. She does wish I could work a little bit more on my own dissertation research, but this year I wanted to work on that, except I just got an idea for another ridiculous paper. I'll keep that under wraps for now. But
You can come back and tell us about it later.
next year maybe. if it'll
Awesome,
get
sounds
published,
good. You can come back on the podcast and tell us all about it.
Yes.
Sounds
So,
good.
anyways, I want to wish you luck with getting your Pokemon phylogeny incorporated into the next Pokedex with Nintendo, and
That'd be nice. Hopefully they won't sue me.
Hopefully this will go well. I think this is the best fan science I've ever seen in my entire real research. So, what do you do with phasmid gut contents?
Right, so my actual research is I studied the digestive system of walking sticks. We have a lot of walking sticks in the museum, and it seemed like it would be easy to study them. Again, this is totally not how you're supposed to do research, but that's how I roll. It turns out that we don't really know much about the digestive system, and there really hasn't even been like many anatomical drawings. The weird thing about all walking sticks, or all that I've seen so far, is they have they've been called appendices coming out of the midgut, which no other insect has, and which I have absolutely no idea what they do. And my advisor does not know, and there's nothing in the literature that suggests anything.
So is this your next quest, is to try and nail down what this
Delve
is going to
into
do?
That's what I've been trying to do in the past, since I finished the Pokemon paper. Is what I'm trying to do now. I've been doing a lot. I've been attacking the phasmid gut in a lot of ways, searching for symbiotic bacteria, searching for any enzymes that might be useful. But then I found these structures, and that's all I found. They kind of resemble the Malpighian tubules, but phasmids have those. If you don't, the kidneys of the insect, Malpighian tubules. But phasmids have those, and these are different, and they don't. What I've concluded through my fall research, and I'll write it up eventually, is these aren't Malpighian tubules, whatever they are. They don't suck things out from the hemolymph. They aren't being used in excretion. So that was the one theory I could find in the literature, and it's wrong. So now I have absolutely no idea what they do, and no idea how to find out.
So
That's
what
awesome.
what kind of approaches are you taking to, like, I'm imagining, like, thinking, we've all dissected grasshoppers or something in some zoology class, and those are like little itty bitty bits. What kind of methods, like what kind of approaches are you taking to try and answer this question?
It's really delicate work, and I don't really have the hand-eye coordination or the teeny tiny tools for it. I mean, I have a little syringe, and it obliterates these tiny little organs because they're all so small. I mean,
How big are we talking here?
We're looking like thinner than a hair. These
wow.
tubes.
Little, all
I mean,
right.
these are about a third. They're about a third to half the Malpighian tubules. So these are tiny little
Wow.
tubes coming out of pretty big insects. So I'm going to try to figure out either how to get really tiny needles and suck out the contents and talk to an analytical chemist to tell me what's inside. But because I don't know whether it's something organic or inorganic, this is way beyond what I've studied. I mean, I'm just a simple organismic biologist.
What have you gotten yourself into, really?
It's
It's a mystery.
frustrating because I'm not equipped to answer this question, but at the same time, it's really exciting because this is an organ that nobody knows what it does. People have forgotten that it exists because it was a mystery to be put aside. So it's really exciting because I'm going to find out something really, really new. I just don't know how to find that out yet.
Do you have any guesses? Like, what is your gut? It's harder to be punny here, but what does your gut tell you about what's going on here? What do you think?
I'm going to check soon to see if they might be harboring microbes. The phasmids don't seem to have symbiotic bacteria, but again, I don't know. I really have no idea what these are. There's something in them. There's a fluid in there. So they do something, but I don't think it's excretion, or if it is, it's really precise. I mean, I'm baffled. This is a little mystery.
That's awesome.
And it's
Mysteries are
mine,
fun.
and nobody else can study it until I'm done.
That's fair. That's
So if
fair.
you've got any ideas on what you think you're doing,
Cut
just
it out.
get out.
Just send it
Cut
to
it.
me and vanish quietly.
Yes, exactly.
Very cool.
That's very cool. And that's the great thing about entomology is you stumble into these kind of things all the time,
Yeah.
and then you completely derailed from what you had originally thought you were going to do. So
I think we can all relate to that on some level. Yeah.
definitely.
Yeah.
That's fantastic. So good luck with that, and
Yeah,
I hope
thank
that
you.
works out for you quite fast before any of these jokers may come up with anything. But I think you
Yeah.
got it. I think you got them now.
Yeah.
So sorry, one more question. Are you doing this with dead pin specimens, or do you have like live specimens you got to do?
Oh no, I just have to be alive.
Right.
I mean,
This
this
is
is a
tribbles,
moving organ, so I need to see what it does in vivo.
So they're alive when you dissect them? You just kind of split them open and...
That's gruesome.
I usually behead them first.
Gently.
All right.
Gently.
Okay, well, there we go. So that's how you do it.
Yeah.
Hooray for no animal care protocols.
Yeah,
It's an invert. It doesn't count.
Yeah,
it
no.
doesn't count, but I try to be nice to them when I can.
There you
Awesome.
go. We appreciate that.
Thanks, Matan.
Yeah.
Thank you very much.
Hi, so we're back here, almost near the end of the conference, and right now we've got Nick Tatarnik from Macquarie University in Sydney, and Nick is semi-famous in Canada. He's been on TV for CBC and with Nature of Things with David Suzuki, playing with tree hoppers in Amazonia. But today, Nick's going to talk about, we're going to ask Nick about traumatic insemination, because I know that Tom is a big fan of stabby cock daggers. And I think he'll get a kick out of this. So,
I see.
And on that note.
Exactly. Over to you, Nick. So, traumatic insemination in plant bugs. How's that go?
Oh, it's quite horrible. Actually. It's males have hypodermic penises, and they stab the female through the abdomen and inseminate their bloodstream. And females have evolved essentially new genitalia on the side of their body to deal with the costs of damage and infection.
So you say that it enters the bloodstream. So how does it get from bloodstream to like where the reproductive stuff actually takes place?
Well, that's a fine question.
I thought so.
Yeah.
Really original.
Yeah, didn't ask this a couple seconds ago.
So the male, the way it works is the male has a hypodermic intromittent organ, and he stabs a female in the side of the body. In some species, there's no modification at all on the female, and in others, they have some sort of a guide or a channel or even a copulatory tube that guides the male to one area where the damage would occur. And underneath that external surface area where they're punctured, whether it's a guide or a tube or something, there's often a big bag of immune cells. And this is basically the immune function of this structure is to clean up the sperm and get rid of any pathogens or garbage that comes in with it. And then the sperm diffuses at the back end of this organ and enters the general bloodstream, so into the hemolymph. And I'm pretty sure in bed bugs they've done some studies where they've sectioned individuals after mating, and they found sperm in various parts of the body, up in the head, leg, and so it sort of goes everywhere. But it does.
It's a problem.
Yeah, it is a problem. But at least
This sounds very inefficient.
yeah, it does. But at least some of the sperm are smarter than others, and they sort of head in the right direction. And in bed bugs, they seem to form an aggregation that heads towards. There's a pair of sperm storage organs called the seminal conceptacles. I think that's what they call them in bed bugs. And some of the sperm heads there, and some of the sperm heads to the base of the ovarioles, and then it passes into the genital tract.
Wow.
And then I guess fertilization occurs as the eggs come down.
This sounds awfully complicated.
It does. It sounds like a waste
This
of
is
time.
that's what I was saying. This is an incredibly indirect route to accomplish something that most other species do much more directly.
Yeah. Well, there's one.
And safely.
And safely, and less stabbing.
Yeah.
Yeah.
Well, there's one bed bug genus where the female has a completely enclosed paragenital tract. So the male stabs the female, sperm goes in, goes through this tube that then goes to, I think it actually goes to the common oviduct. So it goes right to where it would have gone anyway. And then it, does.
They just like to be stabbed.
But with the stabbing.
So-
What's do we know why this might have evolved? Like this, again, it's traumatic for the female. Surely there's some like energetic or health costs there to her.
Well,
What's
yeah.
the point of this?
So it is costly to females. It generally, but actually mating in a lot of animals is costly,
Yeah.
and so elevated mating rates lead to shorter lifespan and often to a lower lifetime reproductive success.
Okay.
In some animals, this is compensated for. It's sort of like the live fast, die young, where the elevated mating rate that also elevates their egg production, and it sort of balances out in the end. But in traumatic insemination, at least studies in the common bed bug have shown that mating rates are about 20 times the optimum for the females in natural populations.
Wow.
So
That's
so
a lot of
they-
stabbing.
So they occur, yeah, so it's not good for them. But having said that, in normally mating things, you get the same thing, and you get comparable mortality and comparable depression of lifetime reproductive success. So I think what that shows us is that the cost mitigation. So the costs are high enough that the cost mitigation evolves quickly enough that the balance is achieved really quickly. And this is what you often see in sexual conflict systems. Is if you just look at a snapshot and you see, females with some crazy structure, males with some crazy structure, but you don't really see any costs in any direction.
Right.
That suggests that the costs have been met really quickly by the counter adaptations.
Oh, interesting.
So with traumatic insemination, what at least I think is happening, and a few other people have suggested this before me, is that it's actually sperm competition that's driving it. So in a lot of insects, you have a simple spermatheca, so a simple storage organ for the sperm. So male mates with female, sperm goes into the bottom of the storage organ, then the next male mates, and his sperm is on the top end. And when the sperm gets used, it's used from the top down, so it's like reaching into a bag.
Yeah.
So that means that the last male to mate is the first to fertilize the eggs.
Yeah.
And if you want to get past that, then you'd want to get past the storage organ. So I think what's happened is traumatic insemination is a way for males to circumvent sperm storage and go straight to the eggs.
And therefore avoiding competition.
And avoid male sperm competition. So although it's a conflict system, it's costly to the females. I don't think the cost to the females is the actual reason for it. Like it's not. The males aren't trying to damage females to suppress remating. I don't think.
Yeah. They're just jumping the line.
Yeah, and that sort of would explain why once a female develops paragenital structures, the male doesn't then try to circumvent those.
Right.
That it's in everyone's best interest if the damage is restricted to one area, because there's some studies that have been done with other traumatic inseminators. If you stab them in
That's
the
the
wrong
best term ever. I'm sorry.
If you stab them in the wrong part of the body, they tend to suffer high mortality.
Well, I was going to ask this, in other organisms, it's a little more random that they'll just stab wherever.
Not so much as stab ear marks.
Well,
Yeah.
So in
Yeah,
so in
no
some
back
bed
marks,
bugs,
but-
there's sort of an area that you just see scars, sort of like a pincushion, in
Wow.
the general area of the body. In the bugs that I'm working on, they've evolved this independently, but they still do much the same thing. But you don't find this really widespread stabbing. It still seems to be restricted to one area. So one of the Australian species that doesn't have any paragenitalia that we can see, the females get stabbed just behind the hind legs, between the thorax and the abdomen. So if you look at a dead specimen and you sort of push apart the body segments, you can see brown melanization. So that's often the only record you have of mating, is these little mating scars.
Interesting.
Yeah.
Interesting. And is there any idea what the cue, is there, so that you're saying there's no visible structure that you can see that would be a cue for, I'm just are there physical adaptations so that they're matching up, that things are getting aligned properly? Do we know anything about that behavior?
Well, for those ones, I've seen them mate, and it's this violent struggle where the males pounce on anything that walks by, and then they try and mate with them. And I guess maybe just the mating position is consistent enough that, and then this is also one of the few parts of the body that's soft tissue. So if they're stabbing and they're hitting cuticle, they won't get through. But as soon as they hit the membrane, they'll get through. And the most accessible intersegmental membrane that they would go through is actually between the first visible abdominal segment and the next one. And that membrane is actually sclerotized in these guys, so that would have been the other place that they would go for, but because it's hardened, they have to move up the body, and that's where they get to this abdominal thoracic area.
That's pretty cool.
Wow. So how common is traumatic insemination? So is it restricted only to the bugs, or are there other insect orders that are going around stabbing things?
So traumatic insemination, you would hope, is rare, and it is rare, but there's actually a lot of things that are doing it. So giant squid do it. Onychophorans, those velvet worms, they do it. Strepsipterans do it, the twisted wing parasites.
Wow.
Anthochorids do it, they're related to bed bugs. There's a couple of other bug groups that do it. There's a nabbit, it's another bug group. They practice traumatic insemination, not through the side of the body wall. They actually enter the genital tract, and then they have a spicule on the tip of their adyagus that punctures the side of the genital tract. The sperm leave the genital tract, parallel that swim along the outside and then re-enter. And yeah, so
It's like the little the commuter lane on the highway.
yeah, exactly. It's the T3 lane. So yeah, so those guys do it, and then I think there's a spider that 2 years ago was found to practice traumatic insemination, and that one it's called Harpacrita sadistica. And if you look up
Aptly named.
traumatic insemination spider on
Yeah.
YouTube, you get an incredible video of this thing. It is just so creepy.
I bet.
Yeah.
Check that
And
out.
then free living flatworms do it, and I think a lot of hermaphrodites do it actually.
Oh, yeah.
I've read that. I don't know if I should say it. Yeah, because I read in a paper that I just accepted. Yeah, that it was a review of traumatic insemination just across the board, and they found that hermaphrodites have a way higher rate of traumatic insemination. Yeah, so lots of these wormy little gooey things seem to be stabbing each other, and and because the hermaphrodites are actually doing each other at the same time.
That's right.
Mutual.
Doing each other, I think, is
Mutual
That's
destruction.
the
Yeah.
is the
technical
technical term.
term.
Yeah.
Yeah. So it's more common. So it sounds like this is a fairly common thing.
Yeah,
Weird.
yeah. And I think it's all about sperm competition.
That's pretty cool.
Yeah.
Not wanting to stay in line.
No.
It sucks for some individuals, but
Yeah, but
Yeah, but see, it's not. It's I guess. I mean, we tend to look at mating systems. You know, we have a human view on things, and we sort of anthropomorphize.
Yeah, for sure.
of course.
But the reality is, these systems. Usually, when you have like a sexual conflict system, you know, typically it's okay. Male has coercive tactics, and females have defensive tactics. But it doesn't seem to be that either sex is actually winning any of these battles because they're cost driven. The costs lead to rapid counter adaptation. So often, you know, like in the bugs that I'm working on, for example, although the females are the ones getting stabbed, they're bigger and they're tough and they kick the crap out of the males. So, you know, so they get a bit of payback, I guess.
Okay.
So it's sort of, I mean, yeah, you can. I feel sorry for all animals, but it's not. Like, I guess traumatic insemination in animals, it's not as creepy as you know it sort of sounds.
yeah.
Oh, it is.
It
It is a little creepy.
That's very cool. So, is there anything else you want to talk about? Life in Australia as a Canadian scientist? Is it much different? We've heard a little bit of Stephen's views on Canadians moving to Australia.
What did he have to say?
He was quite annoyed with the tax system and tax reneging system.
Really, I don't mind the tax system because scholarships are tax free in Australia. I've been audited twice already, but they never caught me, so I got all my money back, which is good.
Very
So
nice.
they seem to give you your money when you claim it.
That's good.
I'm happy with that. It's a bit weird though. Like Christmas time doesn't feel like Christmas. I always feel
Yeah.
a little bit sad when I'm surfing on Christmas Day.
A little
I
bit
feel
sad.
really
It goes
bad
away.
for you. Yeah,
You know,
life
just,
is
you know, when you're
hard.
riding a wave and looking at the sun and it's, you know, the people on the beach and you're thinking, this is not Christmas.
Life is so hard.
Yeah.
How do you cope?
It's tough. It's tough.
But
Other than that,
otherwise, it's a nice place to work, nice place to live.
Oh, it's an awesome place to live. And if you're into entomology, it's just wide open. I think they've described like 23% of their species.
Oh wow.
So
Lots
you
and
can
lots.
go in your backyard and find new stuff to work on.
Yeah, I've
And
heard there's
it's
a lot
not-
of bugs there.
And it's not just they're new and they do weird stuff. So there's lots of ecology and animal behavior and natural history to be worked out. So it's good for that. And that seems to be respected in the Australian scientific community to a certain degree, which might not be everywhere. I mean, taxonomy still gets a bad rap. It gets a bad rap everywhere.
That's normal though, but you know, taxonomists, I don't know.
But having said that, they do have grant programs that actually fund taxonomy exclusive projects. So,
Sweet.
yeah.
Well, that sounds like a pretty good destination for
Sounds
anybody who's
pretty nice.
thinking about it. Plus, they spell words the right way.
This is
Yeah.
true. Yeah, they're one up on. Yeah.
Yeah.
But it's also, Sydney is now either the first or second most expensive city in the world to live in.
Oh.
And they like to charge a lot for everything because they claim that it's, the shipping costs. For example, iTunes, you pay 40% more for everything
Because of all the
shipping costs. Yes.
Wow.
Right.
Yeah.
Yeah, I can see how that works.
And Australia is the only country, as far as at least last year, it was the only country in the world that priced their Mac computers higher than everywhere else. They didn't price it according to like standardized scale.
Wow. So before you move, you should buy all your stuff
Yeah.
first and just bring it with you.
Or buy stuff and sell it there.
Aha.
That's at 20% markup.
Yeah,
Still,
exactly. Exactly.
That's awesome. So when you're back here in North America, you're just downloading songs like mad, aren't you?
Well,
Trying to get caught up on 6 months worth of.
I'm not going to say anything. No, I go to the co-op whenever I come back, Mountain Co-op, and
Cool.
buy a bunch of camping gear.
Yeah, cool. Right on.
Sweet.
That's my story.
Cool.
Cool. Well, thanks a lot for sitting and talking with us.
Yeah.
It was-
I bet you didn't get anything useful out of that.
I bet we got all sorts of useful out of that.
Fascinating stuff.
I think everybody likes to hear a little bit about stabby cock daggers.
Stabby
Yeah.
cocks, yeah.
Yeah.
Oh, I do have to tell you though about my favorite species. I think.
Oh,
yeah.
go for it.
Oh,
One
okay.
more.
Everybody's got a favorite
All
stabby
right.
cock dagger.
So there's one species of Corodromius. These are the bugs with the traumatic insemination, and the male has a corkscrewed intromittent organ, and the female has a corkscrewed copulatory tube on the side of her body. So he actually has to screw into her when they screw.
But
You don't have to.
Yeah.
oh, we'll totally use
You don't have
that.
to
That
use
is the
that.
caliber of stuff we have on this, so
We're
I'm totally
keeping
cool
that.
with that.
That's-
Oh, how many lawsuits do you guys have pending?
That's
Oh,
pretty
none.
good.
I guess you need viewership to have.
Exactly.
That's
Exactly.
it.
So you're good for that one. So
Yeah.
it's all good.
All right. Well, thanks very much.
Thanks, Nick.
Thank you. Thank you.
Good luck with your travels and
Yep.
and various works and tracking down how stabby goes. So
Thanks.
and surfing on Christmas while we're all slugging through the
Yeah, I know
snow.
it'll be tough.
Yeah, we'll think of you.
Thanks.
Not well.
Yep.
Cool. Thanks, Nick. Corey? All right. So we're here with the Bug Chicks at ESA. So everybody's used to the Bug Chicks now on the last episode, and everybody's been super fawning over it. So what are you guys doing here today?
Today we are filming a little short montage video. We're asking people to describe entomology in one word, what entomology means to them. And we're going to blend it all together and see about the diversity and what people think.
And we've got some really cool answers so
Yeah.
far, so it's going to turn out really well, actually.
It's going to
Awesome.
be a good video. Yeah.
That's gonna be sweet to see. So what do you guys think of the conference? I know it was your first time at a big ESA.
Yeah. And yours too.
And
a
mine
big
too,
yes.
yeah. It was
Yeah.
awesome. There are so many amazing talks and not enough time.
No, not enough time to see everything that we wanted to see.
Yeah.
But it was really good to network and to meet people that we've talked to on Twitter for
Yeah.
a
Yeah.
while
and see some of our old friends and lab
Yep.
mates
and get inspired about entomology again. Because
Yeah.
when you're outside of academia a little bit, sometimes you lose it. And
Yeah.
so I just feel invigorated.
Yes.
Yeah,
It's been an
exactly.
awesome conference, ESA. Awesome. Woo!
Definitely recommend it.
Yeah.
So, thanks.
Thank you.
Thanks.
Have fun with your videos and
will.
come back on the podcast sometime.
Yes.
Yes,
Breaking
definitely.
Bio rocks.
Awesome.
It's awesome.
Great. Thanks, girls.